pleasure to introduce Bob Hitchcock. Bob has a fantastically interesting background. I was just looking over his CV and thinking, you know, I'd really like to meet this person. So he started off as a marine seismic technician in Houston, and is now the associate professor of biomedical engineering and director of the graduate program at the University of Utah. Bob's main skill is something we're really interested in. It's the ability to take inventions from the laboratory, particular medical devices, and move it into the real world. And as Mark and I are running a course at the moment, or a teaching block as part of our translational biology, medicine and health program, and that's a message we keep emphasizing. It's not enough to make a great discovery in the laboratory. You have to engage the machinery if you want it to have impact in the real world. Engage that machinery to move it out into the greater world. And that involves interacting with people with skills that you may not be even familiar with. And so I'm sure Bob's going to talk to us about that today. So just briefly, Bob's research interests are biomedical devices, sensors, host response, tissue engineering, bioreactors, and fusion systems. So he's very much into the device area. The other thing about Bob is to consider his context, the University of Utah. We're just discussing how the Mormon community, when they moved out there, they moved out there to get away from everyone because they didn't want to be bugged by everyone. So there's this enormous sense of self-reliance within that community. And there's probably an important ethos which is fused into the entire state, and perhaps part of the reason that there's such a propulsive effort to create biotech companies, technology companies based out of the University of Utah. Sometimes maybe in Virginia we're a little bit self-satisfied, nostalgic, because we've been around for a long time, and this is where the institutions that birthed our country are based. I think we've got a lot to learn from places like the University of Utah. So we're very much looking forward to Bob's talk, and I'll just let you go into it now.
Oh, thank you, Rob. I think I have a lot to learn from visiting Virginia Tech. So thank you for that introduction. I was talking to John earlier today, and he was asking about my transition from industry to academia. And that's where I'm going to start this story today. In 1982, I graduated from the university. I went to work as a mechanical engineer developing instruments that they lowered down into oil wells. And Becton Dickinson hired me in 1988 because they said, you're putting these big sensors into oil wells, and it's not that different from what we want. We want somebody who can put small sensors on catheters and put them in the bloodstream. And so they saw similarities I didn't. Actually turns out that the same materials that make sensors able to survive in the hostile environment of an oil well, these are the exact same materials that we use for long-term implantation in the body. So there are similarities. In 1988, I went to work in the medical device industry. In 2006, for some reason, I left a really good job in the medical device industry and joined academia. I couldn't bring any of my industrial projects with me, and I didn't have a record of collaboration or publication or grant funding, so I didn't know what projects to work on. I developed this very opportunistic, I don't want to call it predatory, but opportunistic behavior, where I started to hang out with clinicians and understand what their problems were. And that allowed me to evolve my lab into what it is today. So some of the first things I worked on were things that I worked on as a graduate student that I thought were really interesting. Cardiac tissue engineering. We had a great project there. Perfused elastomeric scaffolds. You can't do a lot with tissue engineering unless you have some sort of a scaffold. So these elastomeric scaffolds became really important. Physiologic sensors. I was a sensor guy, and so I wanted to work on intra-abdominal pressure sensor, infection control, because this was an area that I knew really well. So this is sort of where I got started, but these led to different projects. So our cardiac tissue engineering and perfused elastomeric scaffolds actually morphed into this intrasurgical cardiac imaging project and imaging catheters, a triple lumen bronchoscopy catheter that we developed for a group in Edinburgh, where we now have a strong collaboration. Esophageal sampling catheter. I mean, all of these came from sort of the same genesis, where we're now sampling esophageal mucus to look at biomarkers for eosinophilic esophagitis. physiologic sensor. So this is done in collaboration with clinicians, and they love to have acronyms for everything. So this is the Wireless Remote Abdominal Pressure Sensor Project, the Maternal and Pelvic Health Project, the Participant Exercise Project, Strenuous Activity Sub-Study, the Pressure Accelerometer Correlation Study, Ambulatory Tibial Load Analysis System. Anyway, it goes on. And then from infection control, we actually started a company in 2008 to make some infection control projects or products. So today I'm going to talk about our abdominal pressure sensing, how we turn this physiologic sensor into something meaningful that's used in people all the time. Intrasurgical cardiac imaging, where we've developed a technology that can be used during open heart surgery on arrested hearts so that clinicians can identify the right type of tissue where they can take suture bites or make incisions. And that involves some FDA approval and an investigational new drug application. And then I'll tell you the story about catheter connections, something that started with a $5,000 micro grant from the University of Utah and was sold in February of 2017 for $40 million. So 3 examples of translational projects that start in the laboratory, sometimes with an inauspicious start, and then turn out to be something important. And this opportunistic behavior that I developed really had to do with finding clinicians who not only had unsolved problems and unmet needs, because they're all over the place, but also the clinicians that could do the heavy lifting, that could help with the brainstorming and help write the proposals and think about the data. So they're a very interesting phenotype, and I'm going to say that not all clinicians are like that. So I was fortunate to be introduced to Ingrid Nygaard. And so Ingrid is a world-class urogynecologic surgeon, and she works on pelvic floor disorders. And what she wanted to do was measure intra-abdominal pressure because it's highly implicated in pelvic floor disorders. So what are pelvic floor disorders? These aren't things that you talk about at Thanksgiving dinner. They're things like fecal incontinence, urinary incontinence, pelvic organ prolapse. It affects almost 1 in 4 women in this country. 1 in 9 women will have surgery for it at least one time, and about 50% of those surgeries, up to 50% of those are revised. And so elevated intra-abdominal pressure has been implicated in the prevalence, occurrence, and recurrence of pelvic floor disorders. We all generate intra-abdominal pressure. And so I want you to think a little bit about how you generate intra-abdominal pressure. We do it when we cough. We do it when we sneeze. We do it when we get out of a chair. Sometimes when we lose the volitional control of our core muscles, we actually generate intra-abdominal pressure in order to stiffen the torso and offload the spine. That's why you see old people getting out of a chair. They grunt sometimes, and that's generating intra-abdominal pressure so that biomechanically they can be a little bit more efficient because they don't have the control in their core muscles. But it's really not clearly understood. The only ways that we had for measuring intra-abdominal pressure were in the clinic. You had to get hooked up to a big machine. You couldn't measure intra-abdominal pressure during normal daily activities. So the problem, there are unreliable methods to evaluate the relationship, and we wanted to reliably track intra-abdominal pressure outside of the clinical environment in order to look at pelvic floor loading and pelvic floor health as a function of intra-abdominal pressure. So here you go. This is the pelvic floor. We all have one. Men have a bladder and a rectum that go through the pelvic floor. Women also have a vagina that goes through the pelvic floor. And so we can measure intra-abdominal pressure directly. Right? So this line's the bottom of our abdominal viscera. And we can measure it indirectly, directly in the peritoneal cavity. So if you're having laparoscopic surgery, they can put in a port, they can measure intra-abdominal pressure.
I think your microphone.
Oh, thanks, Fred. We need to get that on the video, huh?
Yeah.
But we can also measure it indirectly. So we can do that through the vagina, rectum, bladder, and stomach, and people have done this for a long time. Because I come from the medical device industry, we're very familiar with using a design control approach, and so for those of you who have studied it, this is the famous waterfall diagram. And I like to point it out because it does have these loops in it that are really important to close, and that is the verification and validation loop. So verification ensures that your specifications meet your design input. And validation means that the gadget that you finally made at the very end is useful in the hands of the user. So that's validation. And so we had several phases that we used for developing this device, but it's all about documentation and documenting what you're doing. And so our design input document for this project had over 120 different requirements. And so those are requirements for patient comfort, patient safety, the monitoring of the device, the performance, power consumption, radio transmission frequency. Lots of different requirements went into this device. We made them in our laboratory, and this is what the design looks like. So everything here is essentially made in an academic laboratory except for these printed circuit boards. So we mold the elastomeric capsule. We make all of these parts. These are made with a laser cutter and a die stamper, and we operationalize this so that we could support a large clinical trial. So we developed standard operating procedures so that we could design, manufacture, and test. And it's important when you do this in hundreds and thousands of people that you have this type of control, even in an academic laboratory. Because in order to answer the good questions, a lot of times you need a lot of data. In order to get a lot of data from people, you need a lot of devices. In order to control the quality and safety of those devices, you need to have standard operating procedures to design, manufacture, and test these devices. So devices work great, extremely linear. We compare them to the current standard. So the rectal balloon catheter, you can see on the top, rings a little bit at about 22 hertz because it's a fluid-filled system and we have dynamic response. And when you see people jump up and down, you actually get tremendous pressure excursions with a rectal balloon transducer. But with our wireless IVT, except for a little dropout because of sampling errors, we're pretty much rock solid with the reference transducer. So there's a widespread belief that elevated intra-abdominal pressure during certain activities contributes to the incidence, progression, or recurrence of pelvic floor. That's dogma. I mean, that's what people understand. But there's also this unfounded wisdom that after surgery, which 1 in 9 women will get, that you refrain from all physical activity. Why? Well, as Gerton points out in 2008, the only way to protect the tissue at the surgical site from additional injuries is to decrease the pressure in the abdominal cavity, which makes sense. But there are no data to back that up. So after surgery, my wife had surgery, she would say, hey, when can I get back to yoga? 6 weeks. What about swimming? No swimming for 6 weeks. Can I do some Pilates? No Pilates. What exercise can I do? Because exercise is good for me. Helps me sleep at night, helps me digest my food better, decreases my stress. And the doctors say, you can walk and you can get out of a chair, but don't lift anything over 10 pounds. Now that's based on what data? That's based on no data. So my graduate student, Tanner Coleman, just so happened he was dating a Pilates instructor at the time, and he was talking to her about it, and she said, you know, I think some of the Pilates exercises that we do don't raise your intra-abdominal pressure that much. And so he had this hypothesis that, that there were Pilates exercises that women could do after they're recovering from the surgery that wouldn't elevate their intra-abdominal pressure. And so this is what he found. There are 2 types of Pilates that you can do, the mat and the reformer. So the reformer's with the machine and the mat's sort of like yoga, you just do it on a mat. And what he found was that the mean max average of all these Pilates exercises weren't higher than getting out of a chair. And then he looked at it differently. He said, well, maybe it's not about the mean max. Maybe it's about the amount of work and power that it takes. So let's look at the area under the curve. And he found out, in fact, that a number of these activities were higher than the area of the curve. But then he said, you know what, that's not a good way to look at it. Let's normalize this with respect to a normal intra-abdominal pressure, which would be the pressure of somebody walking, which is 40 centimeters of water. So then he reevaluated the data, looked at all of the area under the curve threshold above this nominal 40 centimeters of water, and found out that there were only 2 activities that really raised your intra-abdominal pressure above this. So the takeaway from his paper was that you can do Pilates mat. If you did all the exercises, you would get this number, 12,000. If you did all the reformer exercises you would get 13,000 and a half. And if you put them together, you get 25,000 and a half. And that's the area under the curve, so the amount of work or power that's generated with your intra-abdominal pressure. But if you look at estimated post-op exercise, so this is somebody getting out of a chair 12 times a day and walking for 5 minutes 3 times a day, you see that the area under the curve for their intra-abdominal pressure is actually higher than doing exercises a few times a day. Now, the story is much more complicated than this, but what I want to point out is that when you challenge dogma, when you challenge the clinical dogma with actual data, you can move the needle. And so here was a graduate student who was just like you, working on projects just like you, who had a hypothesis, and that hypothesis resulted in a published manuscript. That manuscript is being referred to now by urogynecologists all over the world in order to help them guide post-surgical activity restrictions in their patients. So that's pretty cool. And how did he do that? He did it through developing a device, providing all of the safety protocols, getting it passed by the IRB, and conducting a number of studies. So he developed this intra-abdominal pressure sensor per AAMI standards. It was used to track intra-abdominal pressure during physical activity. To date, we've monitored intra-abdominal pressure in over 1,000 participants. And this provides evidence to guide post-surgical activity restrictions. So it's possible to use this sensor to understand how IAP plays a role in activities of daily living, and it can be used, these data can be used to help guide post-surgical activity in women who are recovering from surgery. And it's not just a few women, it's 1 in 9 women in this country. So that's a whole lot of people. So where else do we go with this? You know, we started to gather all of these data, and we started to think, gosh, you know, the activities that people are doing are not really prescribed activities. If you ask somebody to get out of a chair, they're going to do it differently than somebody else. And so we started to think about biomechanics. How can biomechanics be used in order to understand intra-abdominal pressure? And so one of the first things that we did was we started to just take a look at the data. What does the pressure look like? So this woman's raising her hand while she walks. Or while she coughs, so we can synchronize the video. But this is your intra-abdominal pressure as you're trail running behind the university. And it's interesting, but it doesn't really tell us a whole lot. And so we started to look at some other activities. Here's somebody powerlifting. So this is a deadlift. What's interesting about this is the maximum intra-abdominal pressure is generated before that bar even leaves the ground. I don't think I could lift this much. And so that gave us some additional insight into what was going on. And so now we actually have a project that's funded by National Institutes of Occupational Safety and Health to look at biomechanics and intra-abdominal pressure in order to better understand workplace ergonomics. Because if intra-abdominal pressure is generated during different activities and elevated intra-abdominal pressure is bad, then maybe we should incorporate ergonomic studies that have to do with intra-abdominal pressure generation in the Ford factory so that when they install transmissions or they put in seats, they can do it in a way that's better for their intra-abdominal pressure. So people had never thought about this before. And it's my example of how you can collect data, visualize the data, and look at it a little bit differently in order to come up with different hypotheses. And so now what we can do with the Vicom motion capture, with force plates, with intra-abdominal pressure, is we can come up with biomechanical models where we can use vectors and moments in order to come up with predictions of intra-abdominal pressure. So now I'm going to tell you a story that's completely different, that has to do with a very talented cardiothoracic surgeon by the name of Aditya Kaza. So AK is an incredibly talented surgeon. He works on infants who have congenital heart disease. And so these hearts are really messed up. If you want to watch a great movie, I think the name of the movie is something God Made, and it was about the first open heart surgery at Johns Hopkins. And these babies are born oftentimes with transposition of the great vessels, meaning that the big blood vessels are in the wrong place. Like your pulmonary arteries where your aorta is supposed to be, you might have 2 valves instead of 4 valves, you're missing a chamber of your heart. And it's up to Dr. Kaza to take these strawberry-sized hearts apart and put them back together. And it's not easy to do. So 1 in 100 babies are born with a congenital heart defect. Most of these defects self-correct. Pediatric open heart surgery is a major therapeutic approach with more than 20,000 of these surgeries performed every year. The problem that we run into is complications due to this conduction system. So sinoatrial node, atrioventricular node, we have the descending branches, Purkinje fibers, bundle of His, and in structurally normal hearts, these conduction pathways are fairly easy to recognize because you can use anatomical landmarks. But in hearts that are congenitally deformed, these pathways could be in a number of different locations. And on the outside of the heart and the inside of the heart, you can't see. You can't see the conduction system. It's not painted green like it is here. And so a precise method for localizing the cardiac conduction system is needed. I mean, it's something that's desperately needed. So we did some studies showing that in non-nodal tissue, so this is non-conductive tissue, we see this regular spatial arrangement of the cardiomyocytes. And in nodal tissue, we see this reticulated, non-organized arrangement of myocytes. We validated that these were, in fact, the conductive or the nodal tissues through anti-HCN4. So HCN4 is a hyperpolarized calcium channel that is indicative of the neuronal phenotype of certain tissues, and it's been shown to be a positive marker in HCN4. So we put 2 and 2 together and said, if HCN4 positive, then nodal cells. Nodal cells are irregularly arranged, no HCN4, working myocardium, and they're arranged very regularly. And so we use that paradigm in order to begin to identify conductive tissue using a very interesting technology. So this is a fiber optic microscope that's developed by a company called Monarchia in Paris, France. And it's meant for endoscopic visualization. And we've adopted it and adapted it so that it can be used during open heart surgery. So we came up with a number of different, so since it's a fluorescent confocal microscope, you need to put dye in there. We can't rely on autofluorescence in order to see the tissue. And so we came up with a couple of different ways of imaging. But essentially what you see here is the way that it's done. You have a handheld probe, you put it on the tissue, you see on a screen what the tissue looks like. So we developed that into a handheld surgical instrument. And that surgical instrument was used in a number of animal studies in order to interrogate these different parts of the heart. And these were some of the images that we got. So on the left-hand side, we see these sort of shaky images of working myocardium. So again, this is a handheld device that's looking at cells at this resolution. And on the right-hand side, we see the sinoatrial node, a regular reticular pattern. And we did this in a number of species in order to prove feasibility. Came up with a method where we had an algorithm that could detect regularity or irregularity. We looked at automated versus human tissue discrimination and published a paper showing that using these 2 methods, Fourier transform and image moments, compared to expert examiners looking at these 2 different types of tissue images, that we could get very high sensitivity and very high specificity with this technique. So this was used to visualize and identify nodal tissue, high sensitivity and specificity. We started this project in 2009. We started with excised rat tissue, then living rat tissue, then we moved to canines, then we moved to sheep, and we've just done our 3rd human patient. So we've just done our 3rd human patient. And so this is a R01 that's funded by the NIH in collaboration with the University of Utah and Harvard Medical School, Boston Children's Hospital. And we have a great team that's working on developing this technology so that it can be used in pediatric open heart surgery. The aim of the grant is to do 60 patients, so we have 60 pediatric patients that we have to consent for this trial and then to look at the data when we're done. So along the way, we thought to ourselves, gosh, we're making this device that's sort of long and skinny, and one of the ways that we access the vasculature in the human heart is through blood vessels. So could we take this device and put it into a catheter? So here's the device, here's the imaging device. This is a sheath, so it's a guide sheath that's used in cardiac catheterization. And here you can actually see the tip of the microscope poking out through the guide sheath in the heart. And so this is a echocardiography of the device in the heart, and these are some of the images that we get. So I said this isn't going to work. So Frank said, we got to try this. I said, look, there's no way this is going to work because we had such a difficult time developing a device that could be held in a surgeon's hand and put on the heart. Matter of fact, we used to play this game, and it was like, who had too much coffee? And you try to hold this thing and try to get images, and it was like, oh, all right, you can't do it, I can, you can't, all right. And so Frank said, no, we're going to put this into a 110 centimeter long catheter. We're going to snake it up into the dog's heart, the beating heart, the blood-filled beating heart, and we're going to see what goes on. And I said, dude, never going to happen. But there was another experiment, and they tried it out, and this is what we saw. And so it turns out that the tip of the fiber optic microscope actually gets caught in the trabeculae in the heart, and actually just goes along for the ride as the heart moves. And when you move it to somewhere else, it gets stuck in the tissue and moves along with the heart. And for me, that was a really important point in my life, because I was, come on, I've been around for a while, I know what works and what doesn't work, and I looked Frank in the eye and I said, this is never going to work. Boy, was I wrong. Looking at blood flow in the heart, in a blood-filled beating heart. And so if that doesn't get you excited, well, you can't work on the project with me there. And what about this? Fibrotic tissue. So fibrosis is a big problem in many diseases, and certainly in cardiac tissue, fibrosis is a big deal. And so what we can do is we can use the same technique in order to image fibrotic tissue. Why is fibrotic tissue important? Because atrial fibrillation is a huge problem in medicine, and the current dogma is that areas of high fibrosis are areas of low voltage. And so if you have low voltage, that's a target for the cardiologist to go, the electrophysiologist to go and ablate. Yeah, but it turns out that that dogma is not true. Based on some of our preliminary data, we're finding that there are low voltage areas that are associated with no fibrosis, and there are fibrotic areas that have fairly high voltage. So we're starting to put all of this together into a story that we can get some funding for looking at this part of the project. So what started as a handheld intrasurgical imaging device is now turned into not only that, because that project's moving along nicely, but also a catheterized version that can go in during procedures and investigate different targets for ablation. In fact, we made one of these and we turned it into an ablation catheter, and we actually used it to ablate cardiac tissue at the same time that we were imaging it, and you can use it as an endpoint visualization for cardiac ablation, which is a big problem. So the last project I'm going to tell you about was a commercial project. And it wasn't my idea, just like the other ones, problems weren't my idea. It came from two lovely nurses. So I'm like, no, they're not that lovely, but they are nurses. And they're not nearly as scary as they look in this picture, certainly not Mike. And they work at the VA, so they're VA nurses, they're infusion specialists. And they came to me and they said, hey, we have an idea for a new device. And it's a device that can be used to protect infusion lines. And I thought, oh, okay, I can probably work on that. And so their idea was this. Matter of fact, this was the very first rapid prototype that I made. So I modeled it up in SolidWorks. I printed these out on the 3D printer. I got an O-ring from Home Depot, and I colored them with Sharpies. And that was it. But here's the problem that we're facing. In infusion therapy today, there are these valves, and they're right here on the catheter side of the infusion line. They're called needleless injection sites, and they're valved catheters. So the male lure goes to the medication reservoir, plugs into the needleless injection site. There's an elastomeric valve in there that gets depressed to allow fluid flow to go through. The Center for Disease Control says these should be maintained with an alcohol pledgette. And you wipe it on there for 15 seconds and all the bugs will magically go away. But we know that hospital patients are susceptible to infections. I mean, it's a huge problem. Direct annual costs of up to $45 billion. 10 years ago, in October of 2008, Medicare said they would no longer reimburse for hospital-acquired infections. They said, that's like bringing your car to get the tires changed, and they wreck the transmission, and then you have to pay for the transmission too. So they're preventable, and they just thought that if they squeezed the money side of the equation, they would go away, and it turns out that didn't really happen. So studies showed that there are a whole lot of nurses that don't swab at all, and that, matter of fact, the CDC recognized that people weren't swabbing enough, and that this was a root of infection. And so you know what the CDC did? Super high-tech solution. They recommended that everyone sing "Row, row, row your boat" while you're wiping this, so that you would wipe it for a long enough period of time. But remember this part, the male lure? So the male lure plugs into the needleless injection site. And so if this is contaminated with bacteria, what happens to that? Nobody was paying any attention to that. Everybody was focused on this. So the solution the nurses wanted was, hey, let's have one of the caps filled with a sponge and some alcohol, and that can screw onto this needleless injection site, disinfecting and protecting it. And the other one is just going to be a dust cap for the male lure. It'll just be a protect against airborne contamination. So I had one of my students go to the hospital ICU when they were changing IV lines, and he clipped 8 male lures. So this is the male lure. This is the top of it right here. So he clipped 8 male lures, and I had him perform SEM on them, scanning electron micrography. And this is what he found. On all of them. So we thought, we just can't make a dust cap for the male lure. We have to come up with an active way of fighting the infection or fighting the pathogens. The problem is it's not a valve. It's open to the medication reservoir. And so how do you protect that? Well, a lot of brainstorming is what happened. So we started thinking about different ways that you could do it, and we had a number of talented engineers coming up with ideas. And it was a simple solution, but it took a whole lot of brainstorming and engineering, a whole lot of pizza. This was every Wednesday night for 2 years, 3 hours every Wednesday night for 2 years. Those were our weekly engineering meetings for this company. We got so sick of pizza that we then got Jimmy John's when they moved into town. We did this for so long, we got completely sick of Jimmy John's too. Then we went to the Corner Bakery or Village Bakery or something. We got sick of their food too. But it wasn't until one weekend that I came in and I developed this device, and I called it the conometer. And what it allowed me to do was test a male lure, and I could precisely manage the translation of the device. I could pressurize it and measure pressure. And I looked at different ways of sealing that male lure. And this was sort of the moment where I was looking through the microscope, and I saw it work. So I saw this small elastomeric cone go into the end of the male lure, seal it off. I saw this foam liberate the alcohol that went on the transverse and lateral surfaces of the male lure and bathe the entire male lure with alcohol and kept it out of the infusion line. And so this was made on a lathe with some UV adhesive. That blue piece of foam right there, I took a biopsy punch and I used a small little sleeping pad foam in order to do it. So if you don't think that you can prototype with stuff that you can get from the lab and Home Depot, you may not be thinking hard enough. So this is the elastomer cone. Here's the residual alcohol that was on there. And we now had a system for disinfecting the male lure as well as the female lure. And so this was it. This was the easy part. You just put a piece of foam in there, fill it up with alcohol. This was the more difficult part. We have reservoir foam, the silicone reaction element, which is a spring, the silicone sealing tip. We had these two little vents on the side of that silicone sealing element. Why do you think we needed those? Because this is a closed system, exactly. Right? So what we found was when we did our accelerated aging tests that we're actually popping the cap out. And so during extended high temperature storage. So, you know, you discover these things along the way, and you have to implement these solutions. So this was the solution. This was the magic right here. And that created the franchise for us. So there was no intellectual property on the female side. I'm just putting a piece of foam with alcohol and screwing that onto a needleless injection site. People had already done that. It wasn't that exciting. But when we discovered and we published on the prevalence of microbes on male lures and how we had a solution that would take care of this, it created an entire franchise for the company. And then our 21-person company fought a huge lawsuit with 3M. So 21-person company versus an 80,000-person company. We spent $6 million on this lawsuit. And so 3M was selling devices, and they knew that they couldn't compete with us, and so their business strategy was to copy our patent. And they did. And they thought that if they just lawyered up enough and if they spent a lot of money on litigation, that we would capitulate and we would license to them, because that's really what they wanted. They wanted license and access to our technology. And it took us 2.5 years. It cost us $6 million, but we prevailed. We got all of our money back from 3M, and they had to pay some damages. Yeah.
Is the intellectual property owned by the University of Utah, or is it within the company itself?
No, the intellectual property was owned by the University of Utah. Yeah, and so the University of Utah licensed it to the company.
Right.
So,
and did the University of Utah help with the lawsuit?
yeah, the University of Utah did help with the lawsuit, but not to the tune of $6 million. Yeah. No, they provided us with, you know, I think more than anything, Rob, they sort of provided us with emotional support. Yeah, knowing that we weren't alone.
Right.
Yeah. And then at some point, there was actually a transaction where the license became exclusively owned by the company just to clean up the relationship a little bit. Yes?
Was that the first time that that happened to you in all of your device?
No,
Or is
no.
that, is that kind of consistent? Is that how you know you have a good idea?
Medical device industry is litigious. And so the engineers that work for me, they get really upset when they have to get deposed by lawyers. You know, I swear to tell the truth, the whole truth, and nothing but the truth. I tell them, look, under deposition, there are 5 answers. Yes, no, I don't know, I don't recall, and please repeat the question. So we go through some training. But here's what I tell them, and here's what I would tell you, too. It's part of the business. It's what we do. And if you don't like it, it's like joining the Army and getting upset when people fire bullets at you, right? It's just what happens. It's part of the job. So 23 patents on this entire system, having a lot to do not just with the method of actuation, but we had to get really clever about how we package these things. So you can go into an ER or an ICU now, and you'll probably see these light and dark blue caps hanging up on IV poles where the nurses can just grab one of these devices with one hand and screw it on the cap. The infection control people, they love this because they can just walk into an ICU and glance around and see that all the lines are protected. And in the hospitals that have brought this product into their clinics, their infection rates have plummeted. So it's interesting to me to think that probably the most impact that I've made in my career is with a 50-cent device. We had an investor who said, you know, I just can't wrap my head around a 50-cent device. So I walked out of the room, came back in with a case of them in a box like this and said, can you wrap your head around a $2,500 box? And he said, yeah, that does me that does me a lot better. So they're manufactured in Utah, right? They're not manufactured overseas. Actually, we did start manufacturing overseas just because of the very low capital expenditure that we needed. But we then brought it back after 18 months to this company, Healthline, in Utah. There are people, you know, it's sort of interesting to me. I mean, here are all the caps stacked up, and this is the warehouse full of them. You go into production and you see stuff like this all the time, just bowl feeders and machines popping these things out, and I call it printing quarters, and they're just popping into here. And so it was a University of Utah opportunity. Again, it started with $5,000, and it was leveraged into a fairly decent-sized transaction. We did have institutional support for this. A lot of talented students worked on this. And so there are students who were actually listed as inventors on some of the patents. World-class local partners, so not only the clinicians, but also the manufacturing, regulatory, and testing facilities that we used. Local economic benefit, obviously. But the most important thing, better outcomes for hospital patients worldwide. And so less infections. So where does this bring us? Those three very, very different projects. And so I think about translation, right? That's what I'm trying to cover in this talk. And here are the different elements of translation that were required. And so all the stuff I talked about was really just innovative and creative and diligent, the stuff that we do as scientists and engineers. But in order to translate, you need some rules of engagement too. And so the intra-abdominal pressure catheter, did it require institutional review board approval? Absolutely. What about approval from the Food and Drug Administration? No. Why? Because the IRB deemed that this was a non-significant risk device. Okay. What about animal testing? No. What about ISO 10993, which is biocompatibility testing? Yes, but only a portion of that. We did a cytotoxicity test on all of the patient contact materials, and so that was deemed sufficient. What about ISO 14971, risk assessment? Yes, we did both a fault tree analysis and failure modes effects analysis in order to develop this device. And we used a portion of the quality system mandated by the FDA, and that's 820.30. For those of you who know it, that is the design control portion of the quality system. And it is, in fact, 820.30 is very important because it's the only portion of the quality system that you need if you want to get an investigational device exemption for your new gadget. So it's important to know. What about on the cardiac side? IRB, absolutely. Did we have to go to the FDA? Yes. They gave us, in conversations with them, they had us do a number of studies in order to get our investigational new drug application approved. So we had to file an IND in order to use topical sodium fluorescein on arrested pediatric hearts. What about animal testing? Absolutely. Toxicity testing, yep, full ISO 10993 compatibility. Same thing with 14971 risk. And yes, we did use a full quality system for this. It was the ISO 13485 quality system. And similarly with catheter connections, we had to engage the full quality system in order to provide these devices to the market for sale. But we didn't require any animal testing unless you consider the bacteria that we tested to be animals. But that was no animal testing required for that. But yes, we did have to do toxicity and risk. And so it's sort of interesting to look at these three very different projects and look at the common denominators here. So the common denominators, doing a risk assessment. Matter of fact, I think a risk assessment is a really important lens that we look through in order to challenge our designs and challenge our assumptions about our designs. And it's a really good, it's a really good way to get away from groupthink also. So risk, important. Knowing ISO 10993 and being able to use that as a means to leverage your products into a translational environment, yes, absolutely. And you have to be conversant with the IRB. And so if you've never filled out an IRB application, it's not daunting. It's just a lot of work. So it's not a lot of intellectual work, it's just a lot of paperwork. Do you need to deal with the FDA? Sometimes no. Matter of fact, many times no. Sometimes you deal with them and it's really straightforward. You have a conversation with them and they say, here are the data we'd like to see, and then you have our approval to move forward. And sometimes you don't. So all of these come with pitfalls, right? Sometimes projects just die. Yes, Rob?
Can you just, in particular for the students here, explain what 510K is?
Okay, so there are two ways that your device can be cleared for marketing. And matter of fact, it's interesting, if you actually read the wording, the only way that the FDA can control medical devices is through interstate commerce. Right? So they're essentially granting you the ability to engage in interstate commerce. So there are two ways that you can get your device cleared for the market. The first is an approval. It's called a pre-market approval. And this is a big deal. And it's typically used for the life-saving, life-sustaining medical devices. If they fail, you die. So we're thinking about like a heart valve would be a good example. Certain high-risk devices are often down-classified into lower-risk devices when we have a lot of experience with them. And one good example is blood glucose monitors. So blood glucose monitors, most successful biosensor in the history of our civilization, were always a class 3 device until we had enough experience with them. And then they were down-classified to a class 2 device. So all of the very risky devices require pre-market approval. There's a scientific review of all of your data and of your facilities, and it typically involves millions of dollars and many phone books worth of paperwork. A much easier way is through a 510K. And so a 510K is your application to the FDA to show that you're substantially equivalent to a predicate device that was made before 1977. And so it's a bit of a loophole. It's a bit of a loophole that people have become very good at exploiting in order to get their devices into the marketplace. But clearly, if you're going to make a new bedpan, should you have a full scientific review of your data and your manufacturing? Probably not. But a bedpan is a medical device, so is a tongue depressor. And so we see at one end these simple devices that shouldn't require a lot of regulatory burden. We see at the other end devices that warrant a lot of regulatory oversight. And then there's everything in the middle. And so there are no real straightforward answers. The best thing that I would recommend for the students in the room is to look at the different devices that are out there, look at their classifications, try to understand why they're classified that way. If you have an idea for a drug or a device, I mean, think about how its regulatory approval cycle might go, what classification of device that would be, and whether there's even a classification for it in the National Registry. All right, so along the way, pitfalls. Sometimes projects die. They do. That's all there is to it. And there are a whole bunch of different reasons. You also have to challenge dogma. As I said to Frank, that'll never work. You're wasting your time. We can barely hold this on an arrested heart. What makes you think that you can put it into a beating heart and get any images at all? And you have to accept a lack of control. So whenever we translate things into the clinical environment, by law, we're not allowed to use these. I mean, I'm not a licensed medical practitioner. And so by law, I'm not allowed to use most of the devices that I develop. And so you have to partner with clinicians and others who can use their device. And that means you have to accept a lack of control. You just have to be able to work with other people. And when you work with doctors, it can be sort of interesting sometimes. So my quote for the pitfalls, there's a thin line between success and failure, and oftentimes you don't know which side you're on. So you should always push as if you're on the failure side, and you want to be on the success side. Caveats. Should always focus on outcomes. Right? So what type of patient outcomes are you going to be looking for with your new technology? Understand the opportunity landscape. So for every project that you're working on, I would say that there's a value proposition and an opportunity landscape. And your ability to understand that landscape and see where the mines are and see where the fruit can be picked, that becomes your opportunity to navigate through that landscape and figure out where your work is. Do the regulatory legwork. There's always regulatory legwork. And I guess in that same bullet point should be the intellectual property legwork. But the regulatory legwork is oftentimes very daunting for those of us who aren't experienced. Interpret the same facts in a different way. So oftentimes the data that you collect from your experiments is meaningful in a way that you don't yet understand. And so learn to look at your data differently. It wasn't until we took those videos of the intra-abdominal pressure that we realized that biomechanics may play an important role in our understanding of the generation of intra-abdominal pressure and how that can play a role in pelvic floor disorders. Same thing with accelerometry. I mean, all of that came from just looking at the same data in a slightly different way. Here's my quote for caveats. Ship in harbor is safe, but that's not what ships are built for. A university in the summertime is awesome without the students, but that's not what universities are built for. And success. So you don't need to commercialize in order to translate. And that's a really important message. And I think it's an important part of us being associated with a university like Virginia Tech and Carilion, that we don't need commercialization. We need patient impact. We need to focus on outcomes. So the intra-abdominal pressure project, we abandoned the intellectual property on our sensor early on in the project because we didn't feel that it was worth pursuing. And so now what are we doing? Even without intellectual property, with no commercialization, with a grant from the NIH, we're publishing manuscripts that are making a difference in clinical medicine. And that's a great investment from society, and we really need to honor that. So laboratories and technical communities are unique ecosystems for translation. I can't say this enough. You know, having come from industry where you might have a good idea, but it gets shot down because it's only going to generate $10 to $12 million in revenue, yet it has an important impact in people's lives. That doesn't happen at the university. You don't get shut down because the patient population is small or the commercial potential is not very big. That doesn't mean there isn't room to commercialize. There's plenty of room to commercialize. But sometimes your ability to make an impact does not have to be monetized. Success brings obligation and commitment. If you don't think it's stressful doing first-in-man open heart surgeries on pediatric patients, then you're probably made of much stronger constitution than I am. But for me, every time my devices get used in people, I get a little anxious. And I want to make sure it's safe. I want to make sure that nobody is hurt with these devices. So success in translation brings this commitment and obligation that you're not only providing a safe medical device or drug or therapeutic to the marketplace, but you're also doing the sustaining engineering that's needed in order to look at adverse events, in order to follow up with your intellectual property, in order to file the right reports, and in order to analyze the data carefully after you collect it. So success brings obligation and commitment. Diligence is the mother of good luck. So the harder I work, the luckier I get. So here's some of the people that actually did the work. I'm just the storyteller here. So a very talented graduate student, Tanner Coleman, who developed the wireless abdominal pressure sensor, and some great clinicians. I can't say enough good things about Ingrid. She is one of the most hardworking and talented clinicians that I've had the pleasure of being associated with. Some great collaborators in our engineering department and funding from the NIH. Cardiac imaging, a lot of people involved in this, and I'd like to highlight two. Adi Ikaza, who is the MD, the surgeon at Boston Children's Hospital, and my colleague at the University of Utah, Frank Saxa. Frank is a very good and close friend and also a very close collaborator of mine, and many of the images that you see in this presentation are a direct result of his efforts. These are the people in Paris that we collaborate with, and the funding that we've received from different sources, but mostly from the NIH. Catheter connections, a couple of great partners, Vicky and Don and Mahender, and then the manufacturing partners. You know, you can't make 1 million devices a week without having a good supply chain. And so we have people who package, people who mold, and people who assemble. And so all of the funding for this, the first $5,000 came from Technology Venture Communications Commercialization Office at the University of Utah. Then they provided another grant of about $50,000, but we raised about $8 million in private capital to get the company going. And that's my story today. I want to thank you all very much for listening to me and letting me tell my story. I'm happy to answer any questions that you might have. Yes?
So you mentioned validating your device kind of after it's out there, and with something like the catheter device, once it's approved and being manufactured and on the system, it seems that kind of your involvement is done. So I'm curious as to how involved or who's responsible for actually testing in the hospital what the reduction in infection rates are and how that works, and if that's something that's required by federal regulations or anything like that, or you just kind of...
So it's an interesting question. Let's start at the end of the question. And that is, do you have to show that you're effective in order to get your device in the hospital? No, you don't. However, we have to make claims on our device. So the claims that we make on our device is that it's anti-infective. And you can't make claims unless you have data to back it up. And so we have shown 4 to 5 log reduction on gram-negative bacteria and some yeasts. And so those are the data that allow us to get the clearance to market our device. But you do not have to conduct clinical trials to show that it's effective. Why? Why? There's only one reason why, and that is because this device is so inexpensive, it's not reimbursed by insurance. And so it's paid for by the hospital. It's just like a 4x4 gauze pad. It's paid for by the hospital. When you're looking at insurance company reimbursement, insurance companies now are getting very, very anxious about showing outcomes. So they're for-profit businesses. They want to show that if they're going to reimburse for a procedure or a device or a drug, that there's some benefit to the patient and actually some benefit to the insurance company as a result. So that's where you do have to. As far as the catheter connections, no. When it was sold and a couple of years ago, I said, and that's just the way it is. I would have been happy to consult for them, but it got bought by a large medical company, and they have the resources to do everything that they want. So I go out to lunch with the guys every now and then, but that's about it. Yes?
So it seems that the 3 companies that you started, at least the problem was validated by a medical professional, whether it's an MD or a nurse.
Yes.
How, I guess, cognizant are they of the problem that exists, and do they have or do you have to probe them? How does that?
Yeah, that's a great question. It's really dicey because you're getting an N of 1 oftentimes, right? Doctors will give you their opinion, and if you want a second opinion, you can ask them again. So oftentimes you have to validate what they're seeing with other people. So, you could work on a problem and they say, gee, that's a huge problem for me, and you talk to another doctor and the doctor says, I don't worry about that at all. And so it's important to do this type of validation. It turns out, so we don't sit down and do an exhaustive interview, right? If we decide to work together, you slowly start to go back and forth about sort of the boundary conditions of the project that you're working on, what some of the priorities might be. It's a lot, and Rob and John, we face this all the time. It's like, who do you collaborate with? A lot of times it's like speed dating. It's like, well, I like you. Okay. All right, let's do that. Can you do the work? Yeah, I can. Can you do the work? Yeah, I can do the work. All right. And so it's really about building these relationships. And I was even talking to Audra about it earlier today, and that is that your ability to network in an environment, meet people, talk to people, understand what different parts of the organization are doing, walk outside the laboratory, learn to use different resources at your disposal in your local environment. And that might even be outside of the Corellian VT enterprise. Your ability to do that in a meaningful way is going to turn you into a more resourceful innovator, scientist, engineer, clinician.
Yeah, okay. So my question was a little bit more when you said you were talking about one of the caveats was you interpret the same facts in different ways.
Yeah.
Do you give credit to that to your ability to have the translatable knowledge in your head, or do you give more credit to surrounding yourself with the right people to do it?
There's no one answer, and I'm sure that many people have looked at the cup half full or half empty or a different way, and it happens to me all the time. So we have a project where we're monitoring the force underneath the foot, and the doctor wanted something so that he could control the partial weight of people who are recovering from tibial fractures. And so we thought we were making an underfoot bathroom scale. Got big funding for this. We conducted a big clinical trial, and we showed that the weight is very variable. We showed that patients behave poorly, and a number of other things. And so that was sort of a couple of years ago. And I was walking my dog, and I was thinking about how I was walking. And all of a sudden it dawned on me that we had three in these cam walkers, these controlled ankle motion boots. We had three sensors, two under the head of the metatarsal and one under the heel. And we had always taken all three of these and summed them up together in order to get the underfoot loading. And I thought to myself, wait a minute, we're not looking at when each one of these sensors becomes engaged temporally. And I thought, you know what? I bet, my hypothesis was that the center of pressure underneath your foot moves to the rear as you get better, as your tibial fracture heals. So if you've seen people walking on crutches or even limping, they're on their toes, right? They're on the front of their foot. And so I asked my grad student to look at all the data and analyze it with respect to center of pressure as a function of time. And we just submitted a manuscript to Gait and Posture that I think is absolutely fantastic. And it shows that you can monitor a person's recovery from tibial fracture by looking at when the peak load is on each one of these sensors. And that at the beginning, when people are first out of surgery, first starting to bear weight, it's all towards the front. But over time, it goes to the back, and you can actually plot the slopes, the rate of change of this, and predict how well somebody's recovering. We saw two people who didn't recover, and I mean, the data are really impressive. You have all these lines that are going down. Those are people who recover. Two people who didn't recover, the slopes are positive. And so that was just, you know, I thought the project was over. I thought it was dead. We had collected all these data. I mean, gigabytes and gigabytes of data. And it wasn't until I was walking my dog and thinking about the way I was walking that I realized that we should take a different look at the data.
So I have a question on behalf of our first-year TBMH students who are undertaking their Mark and I are running a commercialisation blog at the moment with Derek McGarvin, and the students are in the phase where they're searching for their intellectual property to build their pitch around their particular teams. And so we had a get together last night, but obviously the students are very concerned about finding their intellectual property. One of the questions was, what do you do when you don't know what the heck you're doing?
sounds like my job.
Yeah. So I'm wondering, do you have any advice for the students as to where to identify their intellectual property to build their virtual companies?
Yeah, so I guess the question is the slipper looking for Cinderella or is Cinderella looking for the slipper? And so just remember in intellectual property that there's a spectrum of intellectual property that has been actually defined by the courts. And so there are narrow improvement patents where you might just have a tiny little improvement on something that's already known. But then there are pioneering patents that are huge, you know, think airplane, telephone, semiconductor. I mean, these are huge pioneering patents. And so the courts look at these very differently. It's important to understand the way the patents are put together, that the claims at the end of the patent are what that's sort of the fence line for the intellectual property. All the stuff up before that, the preferred embodiment, the specifications, I mean, all of this is nothing more than window dressing to get to the claims. The claims is what, this is what you read. If you need to interpret the claims, sometimes you go to the plain ordinary language in the specifications. But learn to read claims. Learn to read. Sometimes it's legalese, but learn to read them. Also, learn the types of claims that you can make. So you can make plain ordinary claims. You can make what are called 112.6 claims or means plus function claims. And so would they be looking for opportunities where there are no intellectual property, or would they come up with something and then try to figure out how to protect it?
The advice we've been giving is to, say, go to the University of Utah Intellectual Properties Office and look at available technologies.
Oh, for license.
Yes.
Yeah.
And see what's available.
Well, so then you really need to search. So you've got to come up with search strategies. And if you're like me, you just start pounding away at the keyboard. But there's actually better ways to do that. And so you can search by classification, you can search by keyword, you can search by inventor, you can search by company. And so, Google has made it so much easier to search. But I actually find that the United States Patent and Trademark Office website is easier for me to manage than Google Patents. So learn to go to uspto.gov and learn to do, I think it's called the brief search, and you can put in some of the keywords. And remember, patent classifications, so there's an entire classification structure associated with patents. So if I want to look for a scalpel, it's going to be in a certain classification that's very well defined under the broader category of surgical instruments and the broader category of hospital equipment. And it's an environment that you need to become familiar with. It's not daunting, it's not overwhelming. Certainly, it's a whole lot easier than studying for the GREs. And if you can, speak to a few lawyers. Lawyers love to talk. And so yeah, see if you can line yourself up with a lawyer. But spend a lot of time on the website. Play around with it, learn to search, but more than anything else, learn to read claims.
All right, well, if there are no more questions, I'd like to thank Bob one more time.
Yeah, thanks, Rob.